Hi, hello, my name is Anlly Fresno. I'm a graduate student at South Dakota State University. I'm right now doing my PhD over there, and as part of my program, we developed this project where we try to identify what are the main effects of Gray 6. This is a phytobiotic-based product on the lactate producing and the utilizing gut bacterial communities of nursery pigs. So as a brief introduction, with the swine industry still struggling a little bit with the weaning period, and being this still a major challenge for swine producers, especially for the production of post-weaning diarrhea, typically associated with the incidence of enterotoxigenic E. coli, which results in dehydration of the piglets, some retardation, and also sudden death. Industry is trying to develop products that are more naturally for the piglets, and as a result we have developed this plant-based extract additive called or named Gray 6. So the purpose of this project was to evaluate this product to see what potential effect this could have on the bacterial communities of these piglets. So the main objective of this research was to evaluate the effect of Gray 6 on the diversity and composition of fecal bacterial communities of post-weaned pigs. For our materials and methods, we have used a DNA-based approach. So basically, we have extracted DNA from fecal samples that we collected at SDSU in one of our research facilities. After that, after the extraction of DNA, we use a PCR approach to target the 16S RNA gene, which is a very common gene marker used in microbiome studies because it is a gene that is present in all bacterial species, and because it is highly conserved, which means that its sequence has very conserved sections. Now, after we did PCR, we perform a sequence analysis. We send these sequences to a sequence facility, and then we start analyzing these sequences using different bioinformatic tools. One of the processes that we follow after getting the sequences was the cluster into operational taxonomic units. It's a name that we use we designate to a sequence that is a candidate for bacterial species. So we group the sequences into operational taxonomic units, and after that we perform a series of statistical analyses to determine what will be the differences between the treatment and the control group. So as a result, if we go from the broader categories, here we can see a histogram of the general taxonomic profile that was evaluated. In the x-axis, we can see the different groups, the control and the treatment groups. And then in the y-axis, we can see the relative abundance of the different bacteria. Now, green bars and blue bars represents the phylum categories, and then shades within these different colors would represent the different families. So when we look at the general taxonomic profile, we can see that over time we had an increase in the abundance of the phylum Firmicutes, and then we have a decrease in the abundance of the phylum Bacteroidetes. We can also observe here at day 1 that there was a high abundance of the phylum Proteobacteria. Proteobacteria can be characterized or can be recognized for being or for having lots of pathogens in its group. However, we can see that Proteobacteria decreased over time. Now, because we did see some differences between the control group and the treatment group, we decided to go deep in the analysis of this data. So we did an Otu analysis, so these are the results of it. So when we look at the Otu cluster, we decided to take these sequences, or take one representative sequence from each OTU and match it to a database to be able to detect what will be the closest match to a known bacterial species in a database. When we did this, we were able to see that there was one bacteria at day 4 that had an increased abundance when the product was used. This particular bacteria had a percent identity of 98% to Lactobacillus reuteri. Now, this particular bacteria, Lactobacillus reuteri, has been known to have very beneficial characteristics in the gut of different animal species. They have probiotic capabilities, and then it gives the animal some kind of protection in the intestine wall. At day 10, we were also able to identify some increased abundance when we used Gray 6. That was for another Lactobacillus species. In this case was Lactobacillus salivarius. This particular bacteria has also probiotic capabilities in the gut. So this was 2 lactobacillus that were increased by the addition of the product Gray 6. Now, another interesting thing that we observed at both day 10 and day 21 was the increased abundance of this particular bacteria, Megasphaera elsdeni. This bacteria has the characteristics of instead of producing lactate, it consumes lactate. And this lactate, it's transformed into other products such as acetate and butyrate. And these 2 products are very important for gut health because it helps to regenerate the cells of the intestine wall. So with these results, we were able to conclude in this product that Gray 6 can impact the populations of lactic acid and producers and lactic acid utilizers in the gut, which means that it can generate a balance in the gut by the production of bacterial species with probiotic capabilities. So with this, we can conclude that the product Gray 6 can have beneficial effects in the gut of nursery pigs or post-weaning pigs.